Again, welcome to the Artificial Intelligence Precision Health Institute Affinity talk series. And we have a very special talk today that I'll introduce here in a moment. I'd like to start with just a little bit of background. Here we hear from leaders in the field and discuss trends in AI and healthcare, bring together researchers and really a variety of different backgrounds. We never know what the mix is going to be any given time, from computer science, engineering, nutrition, and on and on. Overall goal is to foster collaborative interactions and potentially find new people to work with and solve important problems in healthcare. The format is roughly 30 or 40 minutes for the talk with time for interactive QA. So we like our speakers to leave plenty of time, at least 15 minutes or so at the end for questions. I welcome feedback, and so does Dr. Sadowski, my partner in this effort. So this is brought to you by the University of Hawaii Cancer Center in Kaka'ako, Hawaii. Here is our facility. If you've never visited us, please, next time you're in Honolulu, look us up. Also from the Artificial Intelligence Precision Health Institute is dedicated to training students and faculty in the use of AI for medical research. We have a website you can visit to see some of our projects, and also the Hawaii Data Science Institute at the University of Hawaii, which is cross-disciplinary, also training students in general data science. We have a generous compensation for our speakers, which is a beer stein with our logo etched into it. And believe me when I say that this seems to work. So anyway, our paper of the month is from our speaker, and it's called Generation of Synthetic Whole Body Imaging Tiles of Tumors from RNA Sequencing Data via Cascade Diffusion Models. I have the link here if you'd like to read that on your own. So now it's my pleasure to introduce Dr. Olivier Gevaert. He's an associate professor of medicine and biomedical data science at Stanford University. He's a Belgian-trained engineer and in bioinformatics, and did his postdoc training at Stanford before establishing his own lab. He's been a leader in multi-scale biomedical data fusion, developing machine learning approaches to integrate diverse data modalities that combine molecular, imaging, pathology, and clinical data to support decision-making in oncology. With over 200 publications and a strong record of innovation in computational pathology, radiogenomics, and precision medicine, he's at the cutting edge of this expertise and at the crossroads of AI and healthcare. So please join me in welcoming Dr. Gevaert for his research in cross-modality modeling and digital pathology.
Great, thank you. Okay. Okay, can you see my slides?
Perfect.
Great. Okay. So thank you for that introduction and the kind invitation. Happy to show our work. So I'm going to talk about our work in cross-modal modeling in digital pathology and RNA sequencing data, and show some examples on how we use deep learning and AI models to impute data, and also all the way to generate synthetic data. So my lab, as John was mentioning, is focused on multimodal modeling of patients because we rationalize that each data modality has strengths and limitations. And we get also data from patients, and especially cancer patients, in different forms. There's quantitative features, there's lots of images, different types of images, including pathology images, radiographic images. There's text, we have notes, patient notes that we can get from patients. So all these data come in different forms. Often there's also a lot of missing data. And essentially one of the things that we try to do is to develop embeddings, heterogeneous data banks, lower dimensional embeddings of these different data to use it then in fusion algorithms and use it in downstream tasks. And so that's kind of the topic, and the goal of my lab is to develop multimodal data fusion methods robust methods that can handle various different types of data, different forms of data, also missing data. And we take a lot of advantage of concepts such as pre-training, transfer learning, and in the current context, also foundation models. So this is sort of our vision, is that especially in the context of cancer patients, there's like 4 main data streams or modalities that are relevant for cancer patients. There's electronic health record data. We have patient history there. We have notes, diagnosis labs. More and more molecular data is being collected now routinely for cancer patients, including RNA sequencing, sometimes also DNA somatic mutation panels like Foundation Medicine, or internal assays that some hospitals run. Then we also have digital pathology images. These are high resolution images that pathologists look at to grade tumors and to look at the diagnosis. And then radiology is typically used to follow patients over time and is a non-invasive modality that also has certain advantages. So we focus on developing methods that can handle all these different types of modalities and data streams to then make predictions about patients, about treatment, diagnosis, prognosis, and so on. So this is what we put in this perspective. This is a relatively recent paper from 1 or 2 years ago, where we describe sort of this vision of how you look at all these different modalities for cancer patients, and then modeling of these modalities can be used to extract biomarkers using deep learning methods. So today I'm going to focus on cross-modal modeling. So we're going to focus on modeling RNA sequencing digital pathology data, because in the context of multimodal data, often data sets are incomplete. We don't always have all modalities for all patients. Also, certain modalities are invasive, so we might not be able to gather all these modalities all the time. So modality imputation or cross-modal modeling can be useful because many AI methods and machine learning methods require complete data. So that's how we define essentially cross-modal modeling in the context of imputing one modality from another. And that also means that we're essentially generating partially synthetic data, which also could have advantages in the context of data sharing. So the 2 modalities that I will focus on today are RNA sequencing and H&E images. And so I'm going to show 2 examples. One is imputing RNA profiles from H&E images. And then the second one is generating synthetic images from RNA sequencing data. And then I will end with some perspective on multimodal foundation models. So first, let me give you the example of how we can use high-resolution images to generate RNA-seq profiles. So as I already mentioned, some modalities are more routinely acquired than others. For example, molecular data RNA sequencing is often used as a research tool, but it's not yet routine in clinical practice. But RNA-seq profiles for cancer patients can be essential for subtyping. There's lots of gene expression subtypes known in several tumor types, can be used for drug discovery, for pathway modeling, and so on. At the same time, H&E images, high-resolution images, are the cornerstone of clinical diagnosis for cancer patients. So we always have these images typically available for patients. So the question is, can such an image be a surrogate for an RNA-seq profile? So that's what was our goal. Essentially, can we use that image, and can we use an AI model, a machine learning model that can predict the whole RNA-seq profile? So we looked at a number of tissues, 7 tissues, 7 cancer sites. We used publicly available cohorts from the Cancer Genome Atlas and also normal tissues from GTAGs. If you're not familiar, GTAGs is sort of a similar effort as TCGA, but characterizing normal tissues with RNA sequencing, genomics, and also images are available. And then we developed this model, which is called Sequoia, that essentially uses these high-resolution images and then predicts the whole RNA-seq profile. We use some other data sets for validation. There's the CPTAC cohort, which is proteomic equivalent sort of the TCGA. And then we also had lung cancer data from Tempus. And we also did spatial visualization and validation. We had access to a spatial transcriptomic cohort for glioblastoma patients because the model actually allows us to look at spatial prediction of each of the genes. And even though the model is not trained on spatial data, we can evaluate it spatially. And then secondly, we also wanted to look at how we can, what genes can we actually predict well from these images. So here is our modeling workflow. So we used a vision transformer model to make this prediction. We investigated pre-training of these models with GTACs. And then we also looked at different types of foundation models. We used a typical ResNet, which is sort of the more old way of doing things. And a ResNet is a model that's not trained on medical data, so it's not trained on any medical images, versus a recently developed foundation model by Harvard, which is the Uni foundation model. And then we compared also a number of different models to make the prediction, a simple multi-layer perceptron, and then two types of transformers, including a linearized transformer. So this is the whole pipeline at the bottom. So we have images. Because these digital pathology images are very high resolution, we cannot fit the whole image in memory of a GPU. So we need to create patches. Typically, patches are created of 250 by 250, for example. And these patches are then fed into one of these feature extractors, we can call them, a ResNet 50 or a Uni model. And that creates features. Then, because we know that digital pathology images are heterogeneous, especially of cancer patients, there's lots of different cell types. There's stroma, there's lymphocytes, there's epithelial cells. So we clustered these, and we have up to thousands of patches per slide or per patient. Then we wanted to capture that heterogeneity by modeling the data in about 100 clusters. So we cluster these feature vectors into 100 clusters, and that gives us then 100 clusters that then are being fed into this final module, which are these three versions, a multi-layer perceptron, a transformer, and then a linearized transformer, to essentially model all this data. And then the output of that model is essentially a prediction of up to 20,000 expressions of genes. So now let's look at some results. So I'm going to show two types of results here. So on the left, we have the correlation coefficient between ground truth gene expression and the prediction. So we're going to look at how well the predicted genes are correlated with the truth. And on the right, we can see how many genes we can actually predict with high correlation, with significant correlation, and also low error. So we also look at mean square error to evaluate these models. And then at the top, we have different comparisons. We have the multi-layer perceptron as the final module, the transformer and the linearized transformer, and then each time we compare ResNet with Uni. So let's look at a specific cancer site. For example, breast cancer here in the second column. We see that typically the Uni model always outperforms the ResNet model. You can see the second violent plot always outperforming the left one. And we also see that the linearized transformer is the best model in predicting the number of genes. And you can see that for example, for breast cancer, we can predict up to 18,000 genes with high correlation and significant correlation, also low error just from the image. And then we see similar trends for other cancer sites. For example, colorectal cancer here, glioblastoma. For glioblastoma, you can see the results are lower. We see a correlation between the number of samples we have in each of the cohorts and how well the model performs. So for example, for breast cancer, there's about 1,000 samples available, and this is also the largest cohort in TCGA, and we see better results. So there is definitely a correlation with the data, the number of data points we have. But in general, you can see that in all these cancer sites, the uni model, so a foundation model that was trained on a large cohort of millions of digital pathology images, definitely outperforms a non-medical model. And then here is another couple of sites. So these are lung cancers, pancreatic cancer, prostate, and so on, where again we see the same results. So overall, we did this on 16 different tissues. So now we also wanted to investigate what kind of genes can we predict well, and also how can we validate it across cohorts. So I mentioned that for 7 cancer sites, we had also CPTAC data available. And here you can see how many genes validate in a second unseen cohort. And you see from 43% all the way to 73% of genes are overlapping between the 2 cohorts. So a significant amount of genes are also predicted well in a second cohort. Then we looked at some gene set enrichment analysis to figure out what pathways and gene ontology categories are relevant in the genes that we can predict well. So for example, in lung squamous carcinoma, we see DNA replication. Then we also looked at KEC pathways. And for example, in colorectal cancer, we see genes that are related to cell adhesion molecules can be predicted well from the image. And then we focused more on lung cancer because I mentioned that Tempus cohort that we have access to. So we have a 3rd cohort on lung cancer. And we looked at the genes in lung cancer that we can predict well across all 3 cohorts, so TCGA, CPTAC, and Tempus. And then you can see the genes here in this last plot, and also what pathways they're involved in. And these are sort of the usual pathways that we think are relevant for treatment of lung cancer patients, such as Wnt signaling, apoptosis, cell cycle, angiogenesis, and so on. So this is, I would say, promising in terms of that these models could then be used to get a prediction of the expression just from the H&E image. And finally, so I mentioned already introduction that so we need to use these patches for the digital pathology images because we cannot fit the whole image in memory. But that gives us an advantage that we can also make predictions spatially. So I need to emphasize that the model is trained on bulk RNA sequencing, so it's not trained on spatial data, but we can visualize the predictions for each gene spatially. And we wanted to investigate whether there's any truth to that. And that's what we did here using this Visium spatial transcriptomics dataset on glioblastoma samples from a separate cohort. And each time what I'm showing you is in the left column here is for a select set of genes, the prediction from the model from Sequoia. And then on the right side is the ground truth from the spatial transcriptomic data. And you can see that there's a nice correlation for certain genes and certain samples. And here are some more examples. So each time the first column is a prediction and then the spatial, the ground truth. So we could definitely validate several genes across patients with nice correlation between the ground truth and the spatial prediction. So this is sort of the first example I wanted to give in cross-modal modeling using this vision transformer output. So this work was published in Nature Communications, I believe this year or last year. We also compare with several other models, such as HA2RNA. And then I mentioned this, that we looked at several cohorts, including CPTAC and the Tempus cohort, and the spatial validation in GBM. So let me now go to the second example. So now we also were interested in doing the opposite. So can we create synthetic images from RNA-seq profiles, right? Can we use deep learning models that can use expression profiles of different tissues to generate realistic images that look like real images from these different tissues? And so the rationale is still the same as what I mentioned in the introduction. Multimodal data is often incomplete. We often have modalities that are missing. It might also be expensive to generate certain modalities, especially spatial transcriptomics is a very expensive technique, and we don't expect this to be available for many patients in the future or in the near future. Also, some of these assays could be time-consuming, and some are invasive. Or we have no access to certain modalities because certain publicly available cohorts cannot be linked back to the other modalities. So essentially, we can then again use this cross-modal modeling, but now in the opposite direction to generate, in this case, synthetic images, or we could also still call this data imputation. And generating these type of images may also have advantages in terms of data sharing. Since these are synthetic data, we can more easily share data with outside institutions. So the model that we use for this is inspired based on software tools that probably many of you are familiar with, such as DALL-E 2, and there's a number of other competitors that do similar things, where you can use text to describe something, and then you can generate an image using a diffusion model. And we kind of use the same model for this RNA CDM project that we did. We encoded the RNA-seq profile of 20,000 genes as text, sort of, where then, and we essentially conditioned on that, on those values to generate realistic patches or images of these tissues. So this is motivated based on recent work, diffusion work, where diffusion models can be used in the context of text-to-image outside of medicine. Also, generative adversarial neural networks have been used in the past for generating images. We also have done that, but they have some drawbacks. In particular, we need to train separate GANs for each tissue, and it's harder to develop a model that can do multi-tissue. And so that's why we wanted to use other types of models, such as these diffusion models, because we wanted to create a multi-cancer RNA-to-image model that can preserve cancer-specific characteristics. So we developed a model with a single architecture, so a single model that can generate tiles and patches for essentially 5 different cancer tissues. And then we wanted to evaluate whether the predicted images look like real images using lots of different types of downstream analysis, including, for example, looking at cell types that we extract from these synthetic images, and also can they be used in training machine learning models and foundation models. So these are the 5 tissues that we focused on. So lung, kidney, colon, cervical, and glioblastoma. So we picked these 5 tissues to train this model. Here is the first component of the model. So we first trained an RNA encoder to represent the RNA data in lower dimensional space. So we used quite typical, I would say, variational autoencoder that encodes the data, the gene expression profiles of up to 20,000 genes, for example, in a lower dimensional space of 200 features, where we model the means and variances that then allow us to sample from these distributions and reconstruct the the RNA-seq profile. And this model was actually trained pan-cancer. So we essentially developed this model as a general encoder of RNA sequencing data. And then we use this model in a cascaded diffusion workflow where we have the images, the patches, and then we condition on the latent representation of the RNA-seq data to generate synthetic images with a RNA-to-image diffusion model. First, in the first step, we generate images of 64 by 64, and then we upsample them to 256 by 256, and we can keep doing that depending on how much compute you have. So here are some examples. On the left, I'm showing you some cervical cancer cases, where on the top there are synthetic images and on the bottom real images, and same for colorectal cancer images. We surveyed pathologists blinded to the real and synthetic data, and they could not distinguish real from synthetic patches. And here are similar examples for two other tissues, for glioblastoma and for lung adenocarcinoma. So the big difference we saw in the diffusion model compared to like a GAN model is that we can train a model that can handle all these different tissues at the same time. Versus with GANs, we were not able to, and we had to train sort of separate GANs for lung and brain and so on. Then here is a visualization of the projected RNA-seq data. So in RNA-seq space, so you can see, of course, that RNA sequencing of different tissues is very tissue-specific, and tissues cluster amongst themselves. And then we see synthetic images that are generated that show tissue specificity based on the RNA-seq profiles that we use to generate them. So now, how do we prove that these images are useful or can be used for our downstream tasks? So the first thing that we did is we used a model called HooverNet to identify cell types in these synthetic images, and then we compared distributions of cell types between synthetic and real images. This is for lung cancer, and we saw that there's a nice correlation between tumor cells, lymphocytes, and so on between real and synthetic images. And the same for renal cell carcinomas. So that's like the first thing that we can do is looking at cell types. Are the images representative of real cancer images? Then we also wanted to develop models that can classify tissues, for example, based on mixtures of real and synthetic data. So here I'm showing you a range of using 100% real data up to 75% synthetic data. And we saw no difference in the performance of models trained on real and mixtures of real and synthetic data, but always evaluated on real data in terms of accuracy and F1 score. And then similarly, pre-training and transfer learning are also very common concepts in deep learning and machine learning. We tested several models by including more and more synthetic data, only synthetic data in pre-training a model. And the more synthetic data we use, the better the performance is on a downstream task, predicting, for example, again, tissues among these 5 tissues that we used. And this is true for both accuracy and F1 score. And then finally, focusing on one specific cancer downstream task, which is MSI prediction in colorectal cancer. We found that using synthetic data in pre-training improved the performance. In blue, using more and more synthetic data, we could improve the performance significantly. And we should include more synthetic data because we see still rising performance by adding more and more synthetic data. So that was the published work. We are continuing work on this model, and we have now trained an RNA CDM on all cancer sites, so 33 epithelial cancer tissues. We have now finished that work, and we have added more other cohorts, including a pediatric cancer cohort. And we've started generating images across all these tissues. As you can see here, across a number of additional tissues, and we see nice correlation between each of the synthetic images for each of the new tissues versus the real images. So to summarize, so we developed this cascaded diffusion model with a single architecture that can produce realistic tiles for 5 different cancer types. And we are now in the process of extending this to pan-cancer. Cell fraction proportions are preserved in synthetic tiles, and we believe these synthetic data can be of value to train foundation models, to pre-train deep learning models in combination with real data. And especially this can be useful for oversampling rare tissue types. For example, in pediatric cancers, they are typically more rare. and also other adult cancers such as pancreatic cancer and liver cancers, for example, are more rare compared to breast cancer. And we could use these type of models to generate and oversample more of these rare cases. So how do we put this all together? So in the context of multimodal data, as what I've showed in the introduction, we see that many that patients, especially cancer patients, have often missing data. So not all patients always have all modalities available. And traditional missing data estimation was focused on single modality. For example, in genomics, we often have to estimate missing RNA sequencing data. And we see that now we can go from cross-modal modeling, where we essentially impute a missing modality, all the way to generating synthetic data. We can also potentially generate synthetic data for completely virtual patients in the future. And so we see our work fitting in a broader context of building multimodal foundation models from radiographic images, digital pathology, molecular data, EHR data, that each have their own feature extractors that develop embeddings that then can be used for all sorts of downstream tasks and also generative tasks. They can be used for generate maybe future scenarios, treatment scenarios, and can also be used to make predictions about treatment, diagnosis, and prognosis. So that's kind of our vision for modeling multimodal data, including synthetic data, and building this multimodal foundation model that then can be used for all these for hypothesis testing, essentially. So these foundation models allow to learn the main advantage is that we can learn feature extractors using unlabeled data. So we can use large cohorts for each data modality separately. And then we only need to train a potentially simple fusion layer that is trained on multimodal data. And we may be able to reduce the amount of multimodal data that we need by using foundation models within each data modality. And several of these models are in development, and the field is moving very rapidly in that direction. But of course, we know that there's a computational limitation, and that training these large models with sometimes billions of parameters can be intractable. So that's what I had for today. And thank you for your attention. This is my funding and the people in the lab that did the work. And we also do a lot of work on, in general, multimodal modeling, including radiographic images. And I'm happy to answer any questions.
Olivier, thank you very much. I just want to say I feel really